Welcome to Dr. B.R. Ambedkar Open University. Today we are discussing the BSc 5th SEM Nucleus this topic. Nucleus especially these are the center part of the cell. Actually the first one, the Robert Hooke is the discovered by the cell. Mainly these are the discovered in the bark cells. At the same time the Nucleus also they can discover the in Swedish scientist Robert BRAOU in 1835 is mainly they can discovered in the some of the plant cells, especially in animal cells. What are the importance of the nucleus? Mainly these are the command of the center of the cells. Command of the center of the cells. Nucleus is the why to study of the nucleus? Mainly these are the central part of the cell. Next one, genetic integrity and disease recessive. Disease recessive and the relevance part of the mainly central control of the cell. These are the nucleus govern the cell activities. Mainly they can control by the all the cells. Actually nucleus also present in the only in eukaryote cells. There is no seen into the prokaryotes. Most of the eukaryotes also they are containing the nucleus. These are the nucleus also integrity and control, at the same time function and the development growth of the cell, they all are controlled by the nucleus. Okay. Next one, genetic integrity. Genetic integrity mainly they can control by the safeguards of the DNA and genetic materials, and damage can lead to the some of the it maintains the genetic stability. Next one, disease relevancy. Disease relevance also these are the defects the causes the various diseases, mainly they can use for the genetic therapy. Mainly the nucleus also is the helps the developing of the genetic therapies. Okay. Next one, nucleus. Nucleus also they are containing the four parts. One is the nuclear envelope, the second one is the nucleus, nucleus, nuclear sap, and finally these are the chromatin. Chromatin or chromatin part of the chromatin portion. The first one is the nuclear envelope. What are the importance of the nuclear envelope? Actually nucleus also is the double membrane model. Double membrane model, outer membrane and inner membrane. Inner membrane also they are containing that they can attach resembles to the endoplasmic reticulum. Outer membrane. Inner membrane also there the resembles to the ribosomes. What are the importance of the ribosomes? Ribosomes also they can use for the protein synthesis. Mainly they are using the protein synthesis. Say these are the central control and the genetic integrity and disease relevance. Nuclear envelope. What are the structure and functions of the nuclear envelope? The outer structure, see this the diagram is also outer structure, especially they can white color they can bounded by the nucleus. Mainly is the double membrane model. Most of the nucleus especially they can see into the in prokaryote in eukaryotes, there is no seen to the in prokaryotes. Okay. Main difference between the eukaryotes and prokaryotes, the nucleus, the outer structure also the present the nuclear envelope only they can see into the in eukaryotes. In absence of the nuclear envelope they can see in prokaryotes. These are the major difference the eukaryotes and the prokaryotes. Double membrane model, these are the two lipid bilayer models. These are enclosed, these are the providing the barrier, at the same time they are the protection. Barrier mean especially they can move to the some of the particles, especially RNA, DNA, some of the proteins also they can move into the in barrier structure. These are the major important of the double membrane model. Next one is the protection. At the same time the nuclear envelopes also they can separate the nuclear contents and the shields of the DNA. Especially main nuclear envelopes also what are the major important is the separation of the cytoplasm, and next one is the nucleus. Nucleus also containing the nucleolus or nucleosap, and next one is the chromatin. These are all are present in the nucleus. Mainly nuclear envelopes also they can separate the cytoplasm and nucleus. Okay, nucleus. Next one is the main protection, especially they can the separate of the nuclear contents, the shields the DNA, especially mainly they can nucellus also they are containing the ribosomes, RNAs, proteins, DNA, and different types of the some of the particles in the protein particles also they can see into the in internal part. And next one, regulation, these are also very important. Regulating of the, it control the molecular traffic and regulates the import and export. Mainly these are the controlling regulation, import and export, especially some of the particles, especially the proteins also they can enter into the in nuclear sap. Some of the particles also they can outer structure, especially at the same time nuclear pore also they are present into the in nuclear envelope. Mainly nuclear pores also, what are the major role of the nuclear pores? They can transform the particles, especially one side to another side. At the same time, some of the cells also they are containing the only one nucleus, these are also called as uninucleate condition. Some of the cells also they are containing the binucleate condition. Binucleate means two nucleus also they are containing. Next, so some of the cells also multinucleate condition. Multinucleate means especially they are containing the number of nucleus cells are there containing the multinucleate. These are especially they can see into the in mammals. And next one is the animals, especially animals in the mammal at the plant cells are they are containing the multinucleate conditions. Okay, next one, inner, actually the inner is the envelope also outer structure and inner structure is a double membrane model, mainly resembles to the proteins and the ribosomes. The second one is the nuclear sap. Nuclear sap is also called as the nucleoplasm. Okay, nucleoplasm is the liquid portion. Nucleoplasm also is the liquid portion. These are also they are containing the number of proteins like the DNA and RNA and some of the ribosomes. Number of proteins also they are containing the nucleoplasm. Okay, nucleoplasm. And the next one is the nucleolus. Actually nucleolus, actually nuclear pores also the gatekeepers of the nucleus, especially these are the production gatekeepers, mainly regulating of the molecules passage, facilitate the transport in the inside and the outside, inside and the outside. Next one is the control substances, the traffic condition, the only they can exchange of the essential particles, mainly these are the essential for the function of the controlling the nuclear pores. At the same time, the prevent the pre-movement of maintain a nuclear environment. These are the very important. Mainly they are the prevent the free movement and the maintain the nuclear environment also main important role of the nuclear pores. Next one, nucleolus, already we discussed the nucleus. What are the main important of the nucleus? The rhizome factory. What is the meaning of the rhizome factory? Ribosome factory is the main, is the ribosome is the many protein synthesis, ribosome biogenesis. Then in nucleolus also there are the ribosome biogenesis. Biogenesis means they can formation of the ribosomes. Okay, the ribosome production, the circular protein synthesis, mainly they can form the ribosome production also, one of the major important role in nucleolus. RNA synthesis, mainly RNA synthesis. And next one is the RNA genes are the transcribed here, the essential component of the ribosomes. Next one is the assembly, RNA synthesis, ribosome production. And next one is the assembly, formation. The assembly means they can formation the assembled the some of the protein particles. Ribosomal subunits are the assimilated there, the exported to the cytoplasm. Okay, these are the nucleolus. And next one is the chromatin, especially nucleolus is a new nucleolus also one liquid portion. Next one is the chromatin network. Chromatin network especially they are containing the DNA particles plus some of the histone proteins also. DNA and histone proteins is also called as the chromatin. And next one is the chromosomes. Chromosomes also they can see into the in chromatin particle, especially DNA and RNA, DNA plus some of the histone proteins. DNA main important is the DNA production, DNA nucleus also they are containing the DNA. What are the importance of the DNA? DNA also is are the main genetic heredity. One generation to they can transform the some of the character also the one generation to another generation. DNA and next one is the RNA. The both are the they can see into the in nucleus. DNA and the RNA is the nucleic acids. Nucleic acid also they can see into the in nucleus. What are the DNA? DNA mean deoxyribose sugars, especially DNA is the double helical structure, double stranded helical structure. Okay, next one, these are the discovered by the Watson and Crick model in 1953. DNA also they are containing the mainly the chromatins, especially DNA plus histone proteins is also called as the chromatin. Next one is the DNA also some nucleotides, especially nucleotides mean nucleosides plus some phosphate sugars. These are also called as the nucleotides. Nucleotides mean especially nucleosides plus phosphate sugar. What is the phosphates? Especially nucleosides mean nitrogen bases plus some of sugars. Nitrogen bases plus. What are the nitrogen bases? Nitrogen bases also these are the two types. One is the purines, another one is the pyrimidines. What are the purines? Purines also A plus adenine and the guanine is the both are the purines, and next one is the cytosine and thymine is the both are the pyrimidines. According to Chargaff law, the purines and pyrimidine ratio is also the 1:1 ratio. Okay, main important the DNA particles are the main the genetic variability, especially is the heredity structure. Okay, heredity structure. Next one is the main chromatin also. These are the structure of the chromatin. Especially the structure is the spherical structure. Okay, spherical structure. At the nucleus structure also spherical structure, spherical and the rounded. Especially in chromatin also, especially they can form the in mitotic division of the telophase cells. Mitotic division of the telophase cells. See, this one is the diagram also. Chromatin, especially chromatin DNA is the combined with the proteins is also DNA particles. Next one is the histones. Histones also some of the especially is the four types of the histone: H1A, H2B, H3C, and next one is the four types of the histone proteins. Main important is the proteins which the DNA binds the around proteins particles. Okay. Next one is the mainly histone proteins also. Main important is the role is the some of the proteins. Okay. Next one is the chromosomes. The chromosomes also organize the structure of the DNA. Actually, chromosomes they are containing the autosomes and allosomes. Chromosomes mainly is the chromosome is the chromatin. What is the mean of the chromatin? Chromo mean color like structures. These are also they can see in the nucellus. Okay, nucellus. Next one is the histone proteins. What are main DNA replication also? DNA replication, and next one is the amplification of the DNA also main important role of the in nucellus. And next one is the what are the functions? But at the same time nucellus are the related to the some of the cell organelles like the ribosomes. Next one is the mitochondria, endoplasmic reticulum. Next one is the lysosomes. Actually is the mainly nucellus is all the double membrane model. Only in double membrane model also they can see into the in plant cells and the animal cells. Mitochondria, especially in plant cells are there contained the mitochondria, chloroplast. Okay, in nucellus these are the double membrane model. Okay, next one is the some of the cells especially the coordinate the nucellus also lysosomes also and ribosomes. Main what are the importance of ribosomes also? These are the protein synthesis. Mainly they can use for the protein synthesis, mainly formation of the protein synthesis. Okay, next one DNA replication. What are the main importance of the DNA replication? The copying of the blueprint. What are the copying of the blueprint? Accurate duplication. Duplication means especially formation of the double copies. formation of the double copies, and next one DNA polymerase enzymes. At the same time, nucellus also they are containing the many number of enzymes, polymerase enzymes. Next one is the some of the enzymes main they can key role of the nucleus, and maintaining integrity. Okay, maintaining and integrity also major important role of the DNA replication. Main the DNA replication is the ensures the accurate duplication of the genome. The process also relieves the enzyme DNA polymerase. Mainly DNA polymerase enzyme mainly they can form to the DNA, and the next one is the mainly nucellus they can form to the DNA DNA especially they mainly occurred especially in DNA nucellus, and the next one is the proteins, and next one is the ribosomes, all are the present in the nucellus. Okay, next one transcription from DNA to RNA, mainly RNA synthesis transcription, especially transcription also DNA to RNA formation, RNA to next one is the proteins formation. These are all are the process also is also called as the protein synthesis. Okay, mainly formation of the protein synthesis. Next one at the same time in chromatin especially they are contain the only DNA and proteins, and next one is the next one chromosome and the chiasma also they are present in the rRNA particles. Next one is the ribosomes, mainly ribosomes and rRNA also these are the main important key role is the ribosome synthesis, and next one is the protein synthesis. These are all are the they can correlate to the proteins in the nucellus, and then the RNA synthesis mainly RNA synthesis also they can relate to the ribosomes. In the mRNA synthesis messenger RNA mainly they can transform the particles one place to another place. So some of the particles also they can transform the one place to another places. And the next one is the gene expression, mainly gene expression also how to the genes also they can correlate the in gene expression. The mainly transcription synthesis of the RNA from DNA, messenger RNA mRNA found with the carrying genetic code, gene expression. These are the main key role is the nucleus. What are the nucleus also mainly bounded structure, outer structure is the bounded, mainly the envelope, nuclear envelope also they can protect the nucellus and nucleus, mainly nucleus especially they can see into the in some of the bigger cells also the in plant cells and the animal cells, mainly these are seen to the in eukaryotes. In nuclear envelope also the present of the in eukaryotes, in the prokaryotes also they are absent of the nuclear envelope, maintaining the integrity. Next one is the nuclear matrix. One nuclear matrix nucleus or nucleus app is the neutral matrix. And what is the importance of the nuclear matrix? Organization and support, mainly structure framework, how to form the actually these are the nucleus also one shape they can give the particular shape, rounded shape or spherical shape, and next one the egg shape, different types of the shapes also they can give the structure of the framework. Okay, it provides the shape and support and the organization, mainly based how to especially the size, shape most of the nucleus also shape is the circular shape. Next one is the vernacular shape. Okay, some of the cells also they are containing the only single nucleate shape and double binucleate. In the multinucleate condition also they can see into the in nucleus. Next one is the size also is the minute level size, the millimicron also the size also there is a millimicrons. And next one is the dynamic network. Okay, this is a composed the proteins and DNA regulation function. Main key role is the regulation, central control in it, and the growth and development and function. These are the three characters. These are the characters also main key role of the nucleus. In the essential role also DNA replication and transcription. DNA next one nucellus also they can relate the disease and implication of for the health. For example, approximately nowadays most of the peoples are they can affected by the cancer, number of diseases. For example, sickle cell anemia. okay, anemia, okay, different types of the cells, some of the genetical disorders, mainly these are the genetical disorder also, they can affect by the some of the genomes, some of the especially the particular proteins, for example histone proteins also they can affect the some of the diseases, especially genetical diseases, mainly genetical disease also they can affect the nucleus. Okay, nucleus and disease implication of for the health. See, this one is the approximately cancer, approximately 30 to 40% they can affect of the nucleus also, nucleus. And next one is the some of the genetic disorders, general disorders. Next one is the infections, and next one is the RNC, and next one is the all are the factors they can affect the nucleus. Okay, nucleus. Next one, nucleus mainly the what are the major key role, what are the major functions of the nucleus? Mainly these are the genetic information, mainly genetic information transformation. Okay, what are the genetic information? Actually, genetic information mean these are the nucleus also they contain the DNA and the RNA. Especially, what are the importance of the DNA? The heritable genes, they are the only they can messenger, and next one is that some of the particles, next one is the, some of the information they can transform to the one level to another level, genetic transformation. These are the main DNA also heredity structure. Mainly these are the one generation to another generation, the particles also they can transform the one generation to another generation, heredity structure. ఒక మనం ముఖ్యంగా ఇది ఒక తరం నుంచి ఇంకో తరానికి ట్రాన్స్ఫార్మ్ చేస్తానికి ఉపయోగపడే జెనెటిక్ మెటీరియల్ ఎక్కడ కలిగి ఉంటాయి? ముఖ్యంగా కేంద్రకంలో కలిగి ఉంటవి. మెయిన్లీ ఇవి కేంద్రకంలో కలిగి ఉంటాయి కాబట్టి ఒక తరం నుంచి ఇంకో తరానికి ట్రాన్స్ఫర్ చేస్తానికి న్యూక్లియస్ అనేది మనకు కేంద్రకం అనేది ఉపయోగపడుతుంది. అదేవిధంగా తీసుకున్నట్లయితే స్టోరేజ్ పర్పస్, ఇక్కడ మనకు ఆల్రెడీ స్టోరేజ్ మెయిన్లీ ఎట్ ద సేమ్ టైమ్ స్టోరేజ్ పర్పస్ ఆల్సో దే ఆర్ కంటైనింగ్ ద and nucleic acids, some of the histone proteins, some of the ribosomes. Okay, all type of the storage purpose is the nucleus also mainly is the storage of the some of the particles, especially protein, ribosomes. Next one is the amino acids. Already we discussed the proteins also, main important the structure of the building blocks of the some particles, especially proteins. Next one is the amino acids. Actually, in according to DNA to form to the RNA to protein synthesis, amino acids to the protein synthesis. These are the sequencing of the formation of the protein synthesis. Okay, these are the storage also proteins, amino acids, ribosomes. Next one DNA and RNA. What are the main important? Actually, DNA is the double stranded and the double helical structure. RNA is the single stranded. Okay, DNA is the mainly they can transform the heredity. ఓకే, ఒక తరం నుంచి ఇంకో తరాన్ని ట్రాన్స్ఫర్ చేస్తానికి డిఎన్ఏ ఉపయోగపడితే ఆర్ఎన్ఏ మనకు ముఖ్యంగా ఎక్కడ ఉపయోగపడుతుంది? స్టోరేజ్ పర్పస్లో ప్రోటీన్ సింథసిస్లో ఉపయోగపడుతుందండి. ఈ ప్రోటీన్ సింథసిస్లో ఉపయోగపడేది ఆర్ఎన్ఏ. ఏది ఆర్ఎన్ఏ దేనికి ప్రోటీన్ సింథసిస్ ఉపయోగపడుతుంది? దీస్ ఆర్ ద కోరిలేటెడ్ ద రైబోజోమ్స్. ఇవి రైబోజోమ్స్తోని అసోసియేషన్ చేయటం వల్ల మనకు ప్రోటీన్ సింథసిస్ జరిగి ఇక్కడ మనకు ప్రోటీన్ ఫార్మేషన్ అండ్ నెక్స్ట్ అమైనో యాసిడ్స్ ఫార్మేషన్ అనేది జరుగుతుంది. ఓకే, దీస్ ఆర్ ద స్టోరేజ్. నెక్స్ట్ వన్ రెగ్యులేషన్ ఆఫ్ ద జీన్స్. ఇక్కడ మనకు జీన్స్ రెగ్యులేషన్ కూడా ఎట్లా జరగాలి? హౌ టు కంట్రోల్ ద జీన్స్? ఫర్ ఎగ్జాంపుల్స్ మనకు ఓపెరాన్స్, డిఫరెంట్ టైప్స్ ఆఫ్ ద జీన్స్, ఓపెరాన్ ప్రోటీన్స్. నెక్స్ట్ వన్ ఇస్ ద అమైనో యాసిడ్స్. నెక్స్ట్ వన్ మనకు ముఖ్యంగా డిఎన్ఏ రిప్లికేషన్ జరుగుతానికి ఇవన్నీ కూడా మనకు కోఆర్డినేషన్ చేస్తానికి కూడా మన రెగ్యులేషన్ కంట్రోలింగ్ అథారిటీ అనేది న్యూక్లియస్కి అనేది ఉంటుందండి. అదేవిధంగా తీసుకున్నట్లయితే జీన్ ఎక్స్ప్రెషన్, ఆల్రెడీ వి డిస్కస్ ద జీన్ ఎక్స్ప్రెషన్, ద హౌ టు ఫార్మేషన్ ఆఫ్ ద జీన్ ఎక్స్ప్రెషన్, హౌ టు ఫార్మ్ ద జీన్స్, మెయిన్లీ ద అవుటర్ స్ట్రక్చర్ మనకు ఎట్లా ఎక్స్ప్రెషన్ చేస్తున్నాయి, వాటి యొక్క కదలికలు ఎట్లా ఉన్నాయి, వీటిని అన్ని చేస్తానికి కూడా మనకు న్యూక్లియస్ అనేది ప్రధాన కేంద్రకంగా ఉపయోగపడుతుంది. ఆల్రెడీ రైబోజోమ్స్ ప్రొడక్షన్ కూడా మనకి ఇక్కడ ముఖ్యంగా రైబోజోమ్స్ ఫార్మేషన్. మనకు రైబోజోమ్స్ యొక్క ఇంపార్టెంట్ ఏంది? మెయిన్ ఫార్మేషన్ ఆల్రెడీ మనం చేసుకున్నాం. న్యూసెల్లస్ ఇస్ ద ఫ్యాక్టరీ ఆఫ్ ద రైబోజోమ్స్ ప్రొడక్షన్. ఫ్యాక్టరీస్ ఆఫ్ ద రైబోజోమ్స్ ప్రొడక్షన్. ఇక్కడ మనకు ఫ్యాక్టరీస్ ఆఫ్ ద రైబోజోమ్ ప్రొడక్షన్ తీసుకున్నట్లయితే న్యూసెల్లస్ ద రైబోజోమ్స్ ఫ్యాక్టరీ. రైబోజోమ్స్ ప్రొడక్షన్ చేస్తానికి మనకు ఇలా రైబోజోమ్స్లో కూడా తీసుకున్నట్లయితే మనకు 2 టైప్స్ ఆఫ్ ద రైబోజోమ్స్ ఉంటాయి. ఒకటి 70S రైబోజోమ్, ఇంకోటి అనదర్ వన్ ఇస్ ద 80S రైబోజోమ్. 70S రైబోజోమ్స్లో మనకు ఏ యూనిట్స్ ఉంటాయి, మనకు 50+30 యూనిట్స్ ఉంటాయి. 50 ఇస్ ద లార్జర్ యూనిట్, 30 ఇస్ ద స్మాలర్ యూనిట్. శాటిలైట్కు మనకు రైబోజోమ్స్ ఉంటాయి. మనకు 70S రైబోజోమ్ తీసుకున్నట్లయితే ఓన్లీ ఇన్ ప్రొకారియట్స్లో ఉంటాయి. దెర్ ఇస్ నో సీన్ టు ద ఇన్ ఎస్పెషల్లీ దర్ ఆల్సో దే కెన్ సీన్ టు ద ప్రొకారియట్స్. At the same time, eukaryotes also they are containing the 70S ribosomes and 80S ribosomes. The both are the they can seen to the in eukaryotes. మనకు యూకారియోట్స్లో 2 ఉంటాయి కాబట్టి 80S రైబోజోమ్లో వాట్ ఆర్ ద సబ్ యూనిట్స్ 60+40 సబ్ యూనిట్స్. ఇక్కడ 60 ఉంటుంది, నెక్స్ట్ 40 ఇస్ ద శాటిలైట్ క్రోమోజోమ్స్ ఉంటాయి. ఇక్కడ మనకు ఈ రైబోజోమ్ సింథసిస్ జరుగుతానికి కూడా మనకు ముఖ్యంగా ఇక్కడ మనకు ఆర్ఎన్ఏ మనకు మెయిన్గా న్యూక్లియస్ అనేది ఉపయోగపడుతుంది. అదేవిధంగా సెల్ సైకిల్ కంట్రోల్. ఇక్కడ సెల్ సైకిల్ కంట్రోల్ కూడా మనకు సెల్ సైకిల్ యాక్చువల్గా మనకు సెల్ సైకిల్ జరుగుతానికి కూడా మనకు ఇంటర్ఫేస్, నెక్స్ట్ వన్ ఇస్ ద మనకు మైటాటిక్ డివిజన్స్ ఇంటర్ఫేసు. నెక్స్ట్ వన్ ఇంటర్ఫేస్ నుంచి మనకు మైటాటిక్ డివిజన్ ఎంటర్ కావడానికి కూడా మనకు న్యూక్లియస్ అనేది ఉపయోగపడుతుంది. ఎట్లా ఉపయోగపడుతుందండి? ఈ ఇంటర్ఫేస్లో సెల్ సైకిల్లో చాలా తక్కువ పీరియడ్ ఉంటుంది. దీని నుంచి మనకు ఎక్కడికి మైటాటిక్ డివిజన్లో మనకు యాక్చువల్లీ మైటాటిక్ డివిజన్ సమవిభజన, క్షేకరణ విభజన. సమవిభజన తీసుకున్నట్లయితే మనం ముఖ్యంగా మనకు శాఖీయ కణాంగాల్లో జరుగుతుంది. మనకు సమవిభజన నుంచి మనకు ఏం ఏర్పడుతుంది, ఒక తల్లి కణం నుంచి 4 పిల్ల కణాలు ఏర్పడటానికి ఉపయోగపడుతుంది. అదేవిధంగా ఇక్కడ ఎస్పెషల్లీ ద వన్ సింగిల్ సెల్ దే కెన్ ఫార్మ్ టు ద 4 cells ఫామ్ కావడానికి మనకి ఇక్కడ మెయిన్గా ఉపయోగపడుతుంది. ఇక్కడ ఈ దిస్ టైప్ ఆఫ్ ది మనకు మైటాటిక్ మెయిన్ ద సెల్ సైకిల్ కంట్రోల్. అదేవిధంగా మనకు ఇంటర్ఫేస్ నుంచి మైటాటిక్ డివిజన్ రావడానికి కొంత సమయం పడుతుంది. మైటాటిక్ డివిజన్ నుంచి మనం నెక్స్ట్ వన్ ఇస్ ద మెటాటిక్ డివిజన్. మైటాటిక్ డివిజన్ తీసుకున్నట్లయితే మనకు ప్రోఫేస్, మెటాఫేస్, అనాఫేస్, టీలోఫేస్. మెయిన్గా మనకు అనాఫేస్లో మనకి మెయిన్గా న్యూక్లియోలస్ మనకు అనాఫేస్ మనకు ఫామ్ కావడానికి ఉపయోగపడుతుందండి. అదే న్యూక్లియోలస్లో తీసుకున్న ఫస్ట్ వన్ ఇస్ ద అబ్జర్వ్డ్ న్యూక్లియోలస్ అని ఎవరు ఫాంటానా అనే శాస్త్రవేత్త ఇన్ 1874 దే కెన్ అబ్జర్వ్డ్ ఇన్ న్యూక్లియస్ 1874లో. ఇస్ ఆర్ ద స్పిరికల్ స్ట్రక్చర్. ఎట్లా న్యూక్లియస్ ఎట్లా స్పిరికల్ స్ట్రక్చర్లో ఉంటుంది. ఈ విధంగా మనకు ఎస్పెషల్లీ మెయిన్గా ఇస్ ద మన న్యూక్లియస్ అనేది దెర్ ఇస్ నో స్ట్రక్చర్ ఆఫ్ ది ప్లాస్మా మెంబ్రెయిన్. నెక్స్ట్ దెర్ ఇస్ నో స్ట్రక్చర్ ఆఫ్ ది మెంబ్రెయిన్. అవుటర్ మెంబ్రెయిన్ ఉండదు, ఇన్నర్ మెంబ్రెయిన్ ఉండదు. ఓన్లీ మనకు అవుటర్ అండ్ ఇన్నర్ మెంబ్రెయిన్ ఆల్సో దే కెన్ సీన్ టు ద న్యూక్లియర్ ఎన్వలప్లో మాత్రమే ఉంటుంది. ఎక్కడ ఉండదండి న్యూక్లియర్ మనకు మెంబ్రెయిన్. ఓకే, మనకు సెల్ సైకిల్ కంట్రోల్ చేస్తానికి కూడా ఈ విధంగా మనకు ఉపయోగపడుతుంది. ఓకే, డిఎన్ఏ స్పెషల్ డబుల్ హెలిక్స్ స్ట్రక్చర్ ఇస్ ద హెరిడిటీ స్ట్రక్చర్. డిఎన్ఏ రిప్లికేషన్ అండ్ నెక్స్ట్ వన్ ఇస్ ద రిసెప్టర్స్. రిప్లికేషన్ మీన్ దే కెన్ ఫార్మ్ టు ద నెంబర్ ఆఫ్ కాపీస్. యాంప్లిఫికేషన్ ప్రాసెస్ మనకు పాలిమరేజ్ అనే ఎంజైమ్ ఏం చేస్తుంది, దే కెన్ ఫార్మ్ ద నెంబర్ ఆఫ్ కాపీస్ ఫామ్ కావడానికి డిఎన్ఏ పాలిమరేజ్ ఎంజైమ్ ఉపయోగపడుతుంది. అదేవిధంగా రిసెప్టర్స్, సమ్ ఆఫ్ ద ప్రోటీన్స్ ఆల్సో దే కెన్ అటాచ్ టు ద న్యూసెల్లస్. యాక్చువల్గా మనకు న్యూక్లియర్ ఎన్వలప్లో మనకేంది outer membrane in inner membrane ఉంటాయి. outer membrane these are the resembles the endoplasmic reticulum. ఇన్నర్ మెంబ్రెయిన్ మనకు దేంతో అసోసియేషన్ రైబోజోమ్స్తోని అటాచ్ అయి ఉంటుంది. ఈ విధంగా న్యూక్లియస్ మనకు మెయిన్ కంట్రోల్ పార్టికల్స్, ద కమాండ్ ద సెంటర్ ఆఫ్ ద సెల్స్. నెక్స్ట్ వన్ ఇస్ ద జెనెటికల్ ఇంటిగ్రిటీ అండ్ డిసీజ్ రిలవెన్సీ. అండ్ నెక్స్ట్ వన్ ఇస్ ద double membrane model and protection and regulation. These are nuclear poles are the gatekeepers of the nucleus. At the same time these are the ribosome factory nucleus. These are the chromosome packing of the DNA packing also packing. And next one is the copy of the blueprint, DNA replication and the transcription from DNA to RNA formation. These are all our process are the mainly they are containing the nucleus. These are the nucleus. Okay, finally conclude the DNA and RNAs also they are containing the nucleus. Mainly these are the nucleus also center part of the in the total cell organelles. ఈ సెంటర్ పార్ట్ న్యూక్లియస్ లేనిది ఏ పని జరగదండి. these are the major role of the nucleus. Thank you.